Welcome back to the penultimate talk of Soapbox Science Sydney 2020. I'm Laura McCaughey and I'll be your host for this evening, collating questions and asking our speaker, Associate Professor Michelle Harvey, these questions during her talk. Before we start, I would like to acknowledge the Gadigal people of the Eora Nation upon whose ancestral lands UTSC City campus, from which we are streaming, now stands. I would also like to pay respect to the elders, both past and present, acknowledging them as the traditional custodians of knowledge for this land. Soapbox Science Sydney is taking a different format this year, thanks to COVID-19. Last year, our event was held in person at Circular Quay, where we had over 600 people question, probe, and interact with our researchers while they stood shivering on their soapboxes. Although we've had a change of format, our events still have the same goal at heart, which is promoting dialogue between the public and researchers about topics that are important. On this note, if you have any questions during tonight's webinar, and we hope you have lots, please type them into the Q&A box in your Zoom control panel and we'll do our best to answer all of them over the next 40 minutes. You can ask questions anonymously, and also if you like a question someone else has asked and would like it answered, then please use the upvoting tool, which is the wee thumbs up symbol next to the question itself. This session will also be recorded, but we will not be recording any video or audio input from the audience. If you have any concerns or questions over this, you can contact the Soapbox team via email at soapboxsciencesydney@gmail.com. Our speaker tonight is Associate Professor Michelle Harvey, who has a love of all things insects and has worked in many places as a researcher and academic in forensics, decomposition, and entomology. Michelle's research is gruesomely glamorous, intriguing, and disgusting all at once. She's helping solve crimes and saving limbs too, all with the use of nature's own maggots. Michelle wants to share the use of the natural world to solve modern problems and bring maggots from the lab to the lounge room. I'll now hand over to Michelle to hear all about her fascinating maggot research.
Thanks a lot, Laura, and thanks for everyone for joining me tonight. Maggots, they really are disgusting. People hate them. Maggots are baby flies. They're the offspring, the larvae. They're just disgusting. Flies ruin your picnic, your trip to the beach. Summer is a nightmare because of flies. If you've ever looked in your rubbish bin, you've probably seen maggots in the middle of summer. Probably weren't a fan of these. On your pet food, on the dog poo in the back garden. If you haven't looked, you really should. Insects themselves are everywhere. They're very numerous. They're some of the most abundant organisms that we have on Earth. And blowflies are a really good example of why insects are so successful. As I said, maggots are basically, as you see the cute one here, they're our babies of our blowflies. They're wriggly little things with no legs. Flies are spread all around the world. They live in association with humans and with animals, which means that they get spread frequently on aircraft, ships, in association with domestic animals. It means that fly species are found all over the world. They've spread everywhere. There's around 200 native and introduced species in Australia alone. So blowflies really are a bit of a nightmare, aren't they? Well, I'm hoping tonight I'm going to convince you that that's maybe not just the case. We know that blowflies can do some pretty horrible things. Anyone who knows anything about farming will know that we have an issue with fly strike. So we know that blowflies are attracted to our sheep. They come into what we call fleece rots. So you'll get smelly, urine encrusted fleece on a sheep, and this will bring in the flies. Suddenly you'll have maggots and the sheep become very sick. Sometimes they will even die. In Australia, it costs our economy $280 million a year just in trying to treat and prevent this kind of thing. It costs our farmers dearly. It's an emotional and a labor-intensive burden. But not everything to do with maggots is negative, and tonight I'm going to talk to you about two really important applications of maggots. So forensic science, I'm a forensic entomologist. I'm a blowfly expert. I try to use maggots in a way that they can actually help us and to outthink them on the negative side as well. So in forensics, we exploit blowflies. We know that they're attracted to dead bodies. They love all sorts of distasteful things, and we know that they will come in pretty quickly after a person dies, and soon we will have maggots present on that corpse. We can use these to help us to estimate how long a person's been dead and to narrow down a field of suspects in an investigation. And I'll touch on my research with this later. But I want to talk about something called maggot therapy. Since the Napoleonic Wars, we have known that maggots could clean out gangrenous wounds. In times of war, they were used as nature's own weapon against horrible infections that would have cost people their lives. A fly could strike within a wound, there'd be eggs, suddenly they'd be maggots, they'd eat away that dead tissue and that person's limb would be saved. These were used for a while in medical treatments of wounds, but over time, this tended to kind of die down a little bit because antibiotics became popular in the 1940s. When antibiotics came along, they were that quick pill. They were something that solved the problem. You didn't need to have something wriggling in your wound anymore. We know that maggots do amazing things. When they're crawling around in your wounds, they have these little spines on them, which will irritate the wound, and they cause your body to flush out all the horrible stuff that's present in that wound. The maggot excretes and secretes substances that are antimicrobial. They kill off these horrible bacteria that are causing you so much pain. We know that they also feed on the bacteria and kill them in their stomachs. And we know that somehow, magically, they stimulate the growth of new tissue, which means that your limb can recover. When you have two wounds side by side, one that has maggots in it and one that doesn't, we can see very clearly that on a single human, the maggots have the power to clean the one wound, but the wound that doesn't have the maggots and just has antibiotic therapy just doesn't recover. It ends up needing surgical debridement. We're now faced with a time of antibiotic resistance. This means that we have these bacteria that just don't respond to that magic silver bullet. They don't respond anymore to antibiotics. And what are we going to do about this? How is it that the same flies that cause us so much trouble in fly strike, in hurting and killing off our sheep and costing our farmers, can be used to our benefit in maggot therapy and also in forensic science? So this is something I'm going to talk about tonight in terms of how we can save limbs and lives and solve crimes with our maggots. I'm going to take some questions now. I would love to hear from anyone who has experienced maggot therapy especially.
Thanks, Michelle. I didn't think you were going to win me round tonight, but when you started talking about how maggots can help the problem of antibiotic resistance, you had me sold. I'm all for maggots now. We've got some questions about flies. Why are there more flies in summer?
There's more flies in summer simply because it's warmer. So flies, insects in general, are dependent on temperature. So maggots will grow faster in any kind of environment when it's hotter. You just have to walk outside in the middle of summer and see that there's more insects around. And this comes down with flies, particularly to the fact that in order for adult flies to fly around, they have to basically pump their insect blood through their wings. If it's not warm, they can't do that. And interestingly, even in some of our colder climates, so I'm in Geelong, you will actually find that on a winter's day where you have a real nice splash of sunshine and it's a warmer day than usual, some of those adult flies will come out in the middle of winter. And that's because they spend their winter living on vegetation and hiding away because it's just too cold for them to get out and be active. So the insects are still there during the winter, you just don't see them because they can't be active.
Oh, there's so many reasons I love winter, and that is one of them. You said that the maggots are really bad for the sheep, but they're really good for us. Why is this?
So this largely comes down to the fact that when flies will strike on a sheep, they're not controlled. So I'm going to talk in my next little section about how we do maggot therapy in a controlled way. But we're really careful about the number of maggots we actually apply to a wound in a human. You can't control that in a sheep. So you will have hundreds potentially. On a dead body, we could have thousands of maggots present in one spot. And that burden is just too much. It overwhelms the host.
Oh, okay. So not too many maggots, just a couple of maggots.
Yeah, selective.
So has the maggot therapy for treating bacterial wounds been used in clinics yet, or is it still at the early stages of testing?
It is used. It depends very much on where you are. There's a bigger uptake of it in Europe. There's a number of reasons for that, but they do have some very active commercial laboratories that propose it there. It's used more frequently in the United States than it is here. That said, at Westmead Hospital, they do actually have a maggot laboratory that's producing them. in a way that they're sterile and able to be applied to wounds. So it can be used as a treatment in Australia, and there are pockets of maggot therapy being used. So it is highly recognised as a treatment.
Whenever I talk about antibiotic resistance, I always say what other therapies there are because we're running out of antibiotics. So I'm going to add maggots to my list now when I talk about them. And on that note, I'm just very curious, can the maggots kill lots of different types of bacteria? Or is there some types of bacteria that the maggots can't kill either?
They can be a little bit selective. So without going into lots of terminology, there are differences between different groups of bacteria. So we have something called gram-negative and gram-positive bacteria, which is basically a split that's designed based on the morphology of the bacteria, so their physical characteristics, but can also affect how the maggots react to those in a wound. So we know that the maggots do better with certain types of bacteria, but I guess it's a spoiler alert, we also know that maggots are carrying some very special bacteria inside them that give them the potential to be the superheroes that are going to kill off some of those really bad bacteria.
I'm intrigued to find out more. Do we get different types of maggots, and do we use the different types of maggots for the therapy, or do you only get one type of maggot?
So there's one main type that's used throughout the world. So it's called Lucilia sericata. So it's one of our green bottle flies. It's an interesting species because it also has a sister called Lucilia caprina, and the two of them look pretty similar. They have pretty similar behaviour as well. Interestingly, in Australia, Lucilia caprina is our sheep blowfly, and its close species, sericata, so its sister, is the one that we use in our wounds. But when you go overseas, we see that kind of changes around a little bit. So in some countries, they change places. So one of them will be on the sheep and the other one will be used in wounds. So they seem to be a little bit interchangeable. And I think that might actually be part of the future of maggot therapy, in that, particularly in Australia, we have a heap of species that are native to us. They have evolved to live in our really unique environments through a variety of behavioural adaptations that they have. They can really cope with some hot climates, our unique temperatures in certain parts, and they probably will do better in certain types of wounds. So we know that some of our native species of blowflies are really attracted to things like native animals, like your kangaroos, for example. No one's ever really tested our native species to see how they would behave in a wound. So I think that might be a whole new frontier for maggot therapy.
Oh, very interesting. Somebody wants to know, does the person feel the maggots wriggling during the therapy? Because that would be creepy.
I haven't had maggot therapy, but I wouldn't hesitate to. I think every time I get a wound, I think to myself, oh, maybe this time. I am told that you get a tingling kind of feeling. And that's actually a really interesting question, because they have done studies. So they've done blind studies where they basically said to people, yeah, this is a trial where we're going to look at putting maggots in. So half of you are going to get the maggots, half of you aren't. We won't tell you who's got the maggots or not. And when they've published these studies, it's actually been shown that sometimes the people who didn't even have the maggots said they could feel them wriggling. It's the power of suggestion. We have this ultimate yuck factor with maggots, in that we just imagine that there's something horrible. But when I weigh it up and I think, could I put up with that tingling little tickle in a wound for the sake of keeping a limb and maybe my life? I think I could.
Yeah, I think I could too. I'm sure I'll be lying in bed tonight trying to get to sleep, feeling maggots wriggling all over me. And they're not actually there.
Not literally.
In terms of time, we'll go back to hear a bit more about your research, and then we'll have a Q&A again in a few minutes.
So I'm going to talk a little bit more about the maggot therapy research that's being done in my group at the moment. So anyone who's ever had a chronic wound will know the pain that's involved, and the treatment is quite horrible sometimes when it's a long-standing wound. We're in a time where we're getting more and more diabetic ulcers, for example. It's becoming a real drain on our health system. We have issues in palliative care with bed sores. There's a variety of wounds that really can plague people. And the way that we currently treat them would be antibiotics as the first line of defense, which is fantastic until you get something like MRSA, which is not going to respond to antibiotics. And you need to have another line of defense. So the next way that we would usually try to treat a wound would be surgical debridement. So you would actually go in under surgery and have the tissue scraped out, the dead tissue. And that's obviously something terribly invasive. It requires significant aftercare, potentially hospital stays, or if you can go home, you may need to have frequent visits from healthcare workers to help to continually bandage those wounds. And in most cases with a chronic wound, you may have to have multiple debridements done. So this can take months and months to have that treatment conducted in the hope that you're going to save your limb. What's interesting is that with the advent of antibiotics, we really abandoned what nature gave us. And that's really quite remarkable, given that we know there's lots of organisms in our natural world that have medical properties. We use leeches to help prevent blood clots. We know that bees have antimicrobial properties. That's why when you have a sore throat, everyone says, have some honey, because you know that the honey is going to help you, because bees can help to potentially kill off the bacteria with those antimicrobial substances. So why did we abandon maggots? We know that traditionally in wartime, people would just put in dirty maggots, or flies from the wild would strike their wounds and would lay their eggs, which became maggots in their wounds. We didn't need anything special. We just used what was wild in nature. But suddenly we evolved this to be something highly clinical. We started to use sterile maggots instead. It was like what was out there in nature wasn't good enough. We wanted to have more control over what was happening. But why do we do that? And probably, I'm going to say, the yuck factor. It's the unknown. We were putting maggots into wounds and we knew that it worked, but no one really knew the why. We just knew that it worked. And I talked about the placebo kind of trials, the trials where people didn't know if they had the maggots in them or not. And they all said they could feel the wriggling anyway, because it's the power of suggestion. You mention to someone about putting maggots in their wound and they suddenly get scared that those maggots are going to disappear from their arm inside and they're going to crawl out their nose when they're at lunch somewhere. And so part of maggot therapy and having it become more widely accepted is really education, letting people know what it's really like. So there's been a variety of ways that maggot therapy has been developed. People have developed this tea bag approach. So I've got my tea bag here. Imagine that tea bag was full of maggots. So there's a lab in Europe that actually puts the maggots into the tea bag. And this really deals with that yuck factor, because people then stop worrying that the maggots are going to disappear, because the maggots are contained in this little box inside their wound. We've developed this really nice way that we can create barriers around the wound so that the maggots are constrained. They don't eat too much of the tissue. We are very careful about how many we put in so we don't get too many in there. So we've tried to control the situation so much that we've forgotten about the natural biology of the maggots. What I want everyone to remember is that we all have our own microbiome. The whole reason that we drink our probiotics is that we want to keep our insides healthy, because when you keep your inside healthy, it helps your digestion and it's going to help you and your immune system to fight off potentially infections. So what we do know about maggots is that they have their own microbiome as well. And when we have sterile maggots that we put into wounds, we've taken that away from them. So we take away their ability to do their best in a wound and to help us. So we've been doing research in my lab. I have a PhD student, Nat Gass, has just finished and published on this, in showing that the maggots have their own natural microbiome. They have their own healthy bacteria that live in their stomach. And these bacteria are critical to these maggots being able to kill off harmful bacteria in a wound. And it's quite profound. So a wound is very alkaline, whereas inside a maggot's stomach is very acidic. And we know that some of the bacteria that the maggots naturally harbor only work when you have a lot of acid present in that environment. So they can only really kill off bacteria in a wound when the bacteria are entering into their stomach. So we're really profiling now the microbiome of the maggots and looking at what equips them to deal with a wound that has highly infectious and potentially antibiotic resistant bacteria. And what we want to do is to produce the ultimate maggots. So First, if you had a wound, you would come to us and we would profile that wound. We would look at what bacteria you had present in there that were causing that wound to not heal. What was not responding to antibiotics? Second, we would prepare some nice sterile maggots. So we would make sure that they had nothing inside their stomach. Third, we would then feed them their own probiotic. So we would have maggot probiotics that were designed specifically for your wound to kill off the harmful bacteria that were causing you problems. And then we would put these maggots into your wound and hopefully heal your wound really, really quickly. This is an ideal situation for us where hopefully if we had the evidence basis to do this, we could have commercial labs that would create you custom-made maggots tailored to your own infection. And this is really something that would be absolutely fantastic if it got adopted by governments around the world. And even if your health insurer would say, oh, you can choose maggot therapy as a treatment for that wound. Imagine that. You wouldn't go to hospital, you'd have a maggot clean out your wound instead. And if you think about some of the developing countries around the world that have really limited access to healthcare, particularly in remote areas, this might just provide them with the option to have treatment for a wound where otherwise they may have lost their life.
So many questions about these maggots. First of all, you talked about how we moved away from using wild maggots to using more sterile maggots. Is there the potential that the wild maggots could carry bacteria that would be harmful to humans?
I'd say definitely with what's on the outside of them. Maggots crawl through horrible, disgusting things. And so we always have to wash them to make sure that we're cleaning them. I think what we're looking at the moment is there's the possibility, I think, that they carry certain wild bacteria that are helpful, that are beneficial. Because we've taken a blanket approach to really sterilizing them to make everything as clean as possible and to control everything, we've obviously cut out some of their natural antibacterial capabilities. But I think that there's probably certain bacteria, like you say, that we do need to be removing. But there are some that I think they need to keep because they've learned to live with these. If you think about the relationship between bacteria and antibiotics as an arms race, they're evolving against each other. It's a race to see who can be more successful. Inside a maggot, the whole situation is really different because the maggots are using these bacteria that live inside them. These bacteria are producing their own antibiotics that are fighting harmful bacteria in the wounds for us. So it's almost like taking us from a situation where the scientists can't keep up with the antibiotic race, but we can use the maggots and their own natural bacteria to do it for us. So it's a cheaper and less invasive way to be able to face a really big problem in our healthcare system.
Yeah, the bacteria are always a few steps ahead of us, so it is a really good idea to just use them. How do you sterilize the maggots?
There's a variety of ways that this can be done. The way that we've been working at Deacon in my lab is that we've developed a method for sterile rearing. So it means that we can have nice, clean adult flies that will lay for us eggs, and then those eggs we can wash in a variety of solutions to make sure there's nothing on the outside of them. And then when you feed the maggots, so we feed them up until they get to a few millimeters long before they go into a wound. So you're giving them a kind of head start. And so during this period, you would feed them on a food source that was sterile, just to make sure that you're controlling for what bacteria you are putting back in, because we don't want to be putting anything harmful in. And when these are the kind of flies that naturally live on dead bodies as well, we do have to be pretty careful about what's there.
We've got two questions that are very similar. Do the maggots eat live and healthy tissue if they get out their little maggot pens?
So they could if there were a number of them there. So if you had too many, if you weren't controlling it properly, that could become an issue. So we do actually measure the size of the wound to determine how many you're going to put in there to start with. And it may be that with your little tea bags of maggots, I promise I'm not going to use this tea bag afterwards. That'd be a horrible error if it was full of maggots. With these tea bags, you might be applying a few of them at the same time. to make sure that you can cover the size of that wound, and you create a barrier around the outside. So use a barrier cream like pseudo-creme or some kind of cream that's a little bit less tasteful to the maggots, and then you would tape around the outside to make sure that they stayed in there. And for anyone who's a little bit squeamish and thinks, well, they'd have free-range maggots rather than teabag maggots, which I totally would, we count them in and out to make sure we don't lose any. The free-range maggots are far more successful at cleaning out a wound than the teabag maggots.
Oh, I had a nice little image there of like a little ring fence, like a little circus of maggots and not the cream. So you kind of touched on one of our questions there. Do you put different amounts of maggots for different sized wounds then?
Yeah, so it will depend on the size of the wound, how many you want to have in there, because you need to have enough that you're going to create enough debridement in that wound. It may be that you have to have maybe 2 or 3 courses of the maggots because they're only in there for a few days. A maggot only feeds for, depends on the type, but maybe about 5 days, and then you would remove them. And if that wound still had dead tissue in it, you'd put in a second lot of maggots to help to clean it up.
Okay, that was going to be my next question as well. How long do the maggots have to stay on for? So a couple of days.
Yeah.
Oh, okay. So when you study the guts of these maggots, does it tell us anything about the nature of antibiotic resistance?
So we've only done a little bit of work at the moment in profiling what's naturally found in them. There's a lot to be done still in that space. We've identified maybe 2 or 3 species of bacteria that they naturally carry that we think might be beneficial to them. Our next step is to test this. So we do have a project going on this at the moment.
And could you, with your designer maggots, could you put in genetically modified bacteria that produce specific antibiotics and stuff? Or are you looking more at using natural bacteria?
So we've only looked at the natural because our main motivator was to look at, well, if this has always worked in nature without intervention, what is it they're carrying that helps them to deal with a bacterially rich environment in a corpse or in rubbish? They obviously can survive in those environments very successfully. So we wanted to exploit what was naturally there. I think that if you refine a system where you can custom make these maggots to treat a particular infection, there's no reason why you couldn't go into a situation where you use genetically modified ones.
This sounds really, really cool. Sounds like something out of sci-fi. How long does it take a maggot to turn into a fly? I'm going to assume more than 3 days.
Yeah, so it really depends on temperature. When it's hot, they feed crazy fast. And when it's cold, they tend to slow down a lot. But generalized, if you were sitting around 20, 22 degrees, it'd probably spend a day as an egg. And then maybe about 5 days as a maggot. And then it goes into a metamorphosis, like a butterfly has the chrysalis stage, a pupa, where it's getting all its legs and its pretty wings and everything. And that can take about 2 weeks before they come out.
Okay, I'd like to hear a wee bit more about your research and then we'll wrap up with a longer Q&A in a few minutes. Thanks, Michelle.
Talked about how maggot therapy and custom maggots are going to be the next big thing. But forensic science is also a really important area where we've used maggots to be able to help us with applications that we needed to investigate. So once again, you're exploiting that ability of the flies to be attracted to something that's particularly distasteful. They're attracted to the bacteria. So in forensics, particularly forensic entomology, there are 3 kind of applications that we work with. So stored product is when you might, in your pantry, I think everyone's stored pasta or flour and had weevils and things in there. So the entomologist can be involved in that. Could also be food contamination. If you bought a meal out and about and you had maggots in it and you wanted us to work out how old they were and therefore whether they're in there when you bought it. But a lot of what we do is the medico-legal, also the medical and the legal combination. So we can deal with traffic accidents when a person's driving down the road and they get stung by a bee or a fly flies into their eye and they crash and they claim that's what happened. It wasn't that they were on their mobile phone. We can potentially look at the insect evidence. Cases of neglect. So there have been cases where children have been found suffering from malnutrition and neglect. When they've been taken into the hospital, they're found to have maggots in their anal and genital orifices. differences. So we can determine the age of these maggots as an indication of how long since these children have had their nappies changed, for example. A big thing that we do is the investigation of death situations. So things like the association between a suspect and a death scene. So trying to prove whether a person was in a particular place based on the insects that might be found in association with them. Looking at how a person may have died. So if a person was found with maggots in the palms of their hands, but the body was relatively fresh, this would give us an indication that potentially the person had some kind of wounds to the palms of their hands, because maggots don't have teeth and they can't just bite through skin. The biggest thing we do, and if anyone watches CSI or any of the kind of forensic-y shows, we help to estimate the post-mortem interval, or the time since death. And this is all done on the basis that as soon as you die, your body starts to smell. Once it smells, it attracts flies that come in to lay eggs, which hatch to form maggots, and they move around the body, spreading bacteria that breaks down your tissues into this decomposition soup. And the maggots just drink that up. Sorry for anyone who's eating dinner at the moment, but some imagery for you there. The maggots will feed for several days, and then they will leave the body and they pupate. So they become that kind of chrysalis metamorphosis stage. And by us having a knowledge of how long each of those development stages, the egg, the maggot, and the pupa take, we can estimate how long a person's been dead. So the kind of research that we do, so I work at Deakin Uni. I teach in the forensics program there. So I have a lot of research students who work on different elements of the maggot's behavior that might potentially affect how we estimate the time since death. So there's lots of things that can affect what we call the succession. So when a person dies, and in an ideal situation, they're outside in the sun and the flies get attracted really quickly, we know that there's an order that insects arrive in. So you'll get your blowflies early on, and they have their maggots there. And the maggots are really yummy for other types of beetles and things that come in. And then later on, when the body dries out a bit and it's in that like beef jerky kind of stage, really dry and chewy, you'll start to get other types of beetles in too. So we have a knowledge of which insects arrive at which stage. So the kind of research we do looks at decomposition. And so what kind of factors can affect the order that the insects arrive in and how quickly they grow. So we might be looking at how putting a body in water might affect the insects. Will the insects still come? Will you still get maggots present? Will they grow at the same rate? What happens if a body's buried? Do you still get the insects coming and colonizing it? Can we still use the maggots to estimate the time since death? What if a body's covered in vegetation? A kind of popular way to be able to conceal remains quite quickly without digging a hole. A big area that we work in is a multidisciplinary one between chemistry and entomology. We do entomotoxicology. So if you imagine that a person dies and they have some kind of poison or drug present in their system, we can actually use the maggots to tell us what was present in there. Because later on in decomposition, when there's not a lot of tissues left or a lot of body fluids that you can test for toxicology, we can actually use the maggots themselves. And so we can detect if there are drugs present in these maggots. And some of these maggots, when they feed on certain types of drugs, like cocaine, for example, might actually feed a lot faster. So there was a case where they actually pulled a maggot from the nasal passages of a person who had cocaine residue in their nasal passage. And so that maggot was absolutely massive. It had grown faster than any other maggot. So we look at how the presence of different substances can affect the growth of the maggots and whether it affects what order the insects come. We also look at a common way of, I guess, concealing remains or trying to remove biological evidence might be to cover a body in bleach or lime. Or clean it with disinfectant. And we look at how these factors can affect the decomposition of the remains. And even some of the things that a person might incidentally just die with on their surface. So sun cream, insect repellent, perfume, these all affect how the insects are attracted to you after you die. So we look at how they affect how quickly those insects will be there and how they affect the maggot's growth. So we have a lot of that kind of work that goes on, and it just shows that maggots have this amazing ability to live in a wide variety of environments and to deal with these areas that have so much bacteria. They might have lots of substances that are really distasteful and disgusting, But maggots have developed the ability to live in areas that we couldn't possibly deal with. The amount of bacteria that they can withstand and quite often kill off is actually quite phenomenal. And so we've learnt to capitalize on this in forensic science. We use their attraction to smelly things to our own benefit. We're doing the same thing at maggot therapy as well. We're exploiting their ability to clean up environments that have too many bacteria in them. So this kind of shows you that maggots really are our next best friends. They're not something that's always distasteful. But how can we turn this around and also conquer fly strike? Our farmers in Australia are suffering and have for many years as a result of these flies attacking their sheep. Is it possible that we can now use this knowledge of how flies interact with bacteria to be able to conquer fly strike, to stop this from causing such a massive economic loss in our agriculture sector? It's quite possible. Something so disgusting as a dead body, it's so amazing that a maggot can be used to help to solve crimes, that we can use it to clean up a wound, that one day you might walk into your doctor's office, that your doctor will swab your wound, send away for a custom set of maggots to treat your particular wound, and your health insurer might even pay for it because it's recognised popularly as being a routine treatment for chronic wounds. And this would keep people out of our hospital beds as well. Maggots aren't fazed by antibiotics. You can have antibiotics at the same time as you have maggot therapy. They remove the need for surgery. They're cutting down treatment time in hospitals. And it's just less invasive and less pain for the patients. It overcomes that whole, I refer to the arms race between bacteria and maggots. So the maggots are harboring these bacteria that are able to make sure that there are antibiotics that are there to treat harmful bacteria in our wounds. So we're really harnessing what nature has given us in a blowfly and their association with bacteria to our own benefit. So when you look around and you look at the dog poo, or you look in your garbage bin the night before the garbage gets collected, you look at those maggots and you think that's absolutely disgusting and that stinks. Stop for a moment and appreciate the environment that they're living in. They've evolved a way to be able to deal with something that's potentially so harmful to a human being. They can live in that environment, and they have the potential to be able to be applied in a hospital setting or just in a clinical setting and be able to save limbs and to save lives. And I guess from my perspective, I have to end by saying, who wouldn't want a custom-made maggot?
I think if my custom-made maggot was the wee fluffy cuddly one that you've been showing us, I'd be all for that. I'll start with a random question. Could you trick the forensic officers by having maggots that are a few days old, drugging them up and then throwing them on the body you've just killed?
Potentially, but it's more likely that when we looked at the situation, we could also use the stage of decomposition of the body itself to help to tell us that potentially something wasn't quite right here. So it wouldn't be unheard of that maggots were planted. More commonly, that would happen in a food contamination situation, but it could happen in a death situation. But we would be able to look at the condition of the remains and pretty much know that something wasn't quite right.
Okay. What about the oxygen and pH ranges that maggots can tolerate?
There's not a massive amount of information on that. We do know that they need oxygen. So I should say maggots breathe through their bums, if anyone didn't know this. If you can see the bottom of my maggot here, you can see these two round little circles. So maggots actually breathe through their butts, and that's because when they feed, they'll all line up with their pointy end down, which is their head, and they have these little mouth hooks, there's no teeth, and they all have their bums lined up at the top, so they can keep breathing constantly while they're feeding. They need to be able to take in oxygen, and that's why we don't have to worry about maggots crawling inside us and disappearing during maggot therapy, because they just couldn't survive in that situation. We know that they deal really well with alkaline environments because that's what a wound naturally is. I haven't seen a lot about how acidic the environments are that they can go into, but it doesn't really affect us for maggot therapy. they are incredibly resilient, even with the temperatures that they can withstand. One of the first things I saw when I was doing human decomposition research was that I encountered a set of human remains where it was a pouring rain day and it was about 20 degrees, the ambient temperature, but there was steam coming off this body because there were so many maggots on there. And the maggots, they mass together, they group together, and it keeps them all nice and warm, and they feed very effectively there. And the temperature of this bunch of maggots, and a couple of thousand of them there, was 65 degrees.
Wow.
So they're what we call exothermic. They produce lots of heat. So not only are they living in this terrible squalid environment filled with bacteria that would kill most of us, they can also deal with some amazing heat extremes as well.
I was just about to ask you what's the most disgusting thing you've ever came across in your work, but if it's not that, I don't think I want to know the answer.
Actually, I don't think it would be human decomposition. So a lot of my research has been human decomposition research at the decomposition facilities in the United States. So I have watched the full decomposition from day one for several months on a daily basis, looking at these remains. That doesn't disturb me. I think the worst thing I ever smelt that really turned me off, it's probably dead animals more than humans. People always ask what the smell of human decomposition is, and it's something that you never forget. But I find the smell of rubbish, poo, or dead animals far worse.
Okay, I'll have to take your word for it because hopefully I'll never smell a dead body. What have you? Am I right in believing there's a body farm has opened in Australia, and have you been to that body farm to further your work?
Yep. So there's one in Sydney. Well, near Sydney. So this is one that's hosted by UTS. So it's an amazing facility. It's called After. They're doing some fantastic research there. I'm not doing human research there at the moment. So I've got my own entomology research happening here at Deakin in Geelong. So I'm in Victoria. But most of my work's been done at the Anthropology Research Facility in Tennessee, and also one at Texas State Uni.
And one last question, and this is a nice question, and hopefully you don't give a disgusting answer to this as well. What got you into science, and what makes you love it?
So when I was a kid, I was the kid who sat there and poured water down ant nests and wanted to see what the ants did. And I was chasing butterflies around from the time I was about 6. And that was always my passion. I never thought I'd do that for a job. I got to uni and I just didn't know what I wanted to do. So I did a Bachelor of Arts and a Bachelor of Science. So I was studying Chinese language and entomology. And then I had one lecture on forensic entomology, and I just thought, this is pretty interesting. And I knew I wanted to do something that was going to help people with science, particularly with entomology. And so I decided that I'd move into forensic science and see how I could use insects in that way. And so my first time I ever saw a dead body, and this is not disgusting, was actually a child. And while that was, something that was difficult to take, it was the most reinforcing experience of my career, because at that moment I knew that I was using objective science to be able to potentially bring about a successful conclusion in a case, and to bring peace of mind to the family of that child. And that's an incredibly powerful thing to be able to do with science. I always say to people, every day I get up and I get to go to work, and I'm an academic. I get to run a research lab and find out cool stuff every day. I get to travel, obviously not at the moment, and I get to teach. And the teaching is the really fun bit, because, research is great. You find out new things and you can help people. But when you teach, you get to see a student's face light up, and you get to see that spark there as you show them something that they hadn't imagined before, and it can spark in them a new dream. And so that's even when things are really tough and there's lots of admin on you, when you teach a student and you see that in their face, that's enough to show you you're doing the right thing and that you've made a contribution in life. So For me, science is about doing something I absolutely love, but being able to help people by doing it.
Thanks for that, Michelle. That really was a brilliantly disgusting talk. And thank you to everyone who attended and asked questions. If you want to watch this talk again, or you know someone who would have enjoyed it, then we'll be posting it on our website in the next few weeks. You'll also receive a post-event email in the next 24 hours with an evaluation survey. Please fill this out as it helps us to gauge the success of our events and make improvements for next year. We couldn't run Soapbox Science Sydney without our very generous sponsors. So I'd really like to thank the University of Technology Sydney, Excite on Science, Equus, and the University of Wollongong for their continued support. We've got one more exciting talk lined up tonight, and it will be starting in 15 minutes with Julianna Kedar talking about her research into shark tracking. You can join this talk from the link you're on now, or you can find more information on our website, soapboxsciencesydney.com. Thank you again for attending, and we hope to see you again in 15 minutes.